SARS-CoV-2, the virus that caused COVID-19, probably originated in late 2019 in China. Quickly, the virus had spread to over 200 countries all over the world, and by mid-March was recognized as a global pandemic. Scientists were able to determine the structure of the virus and how it infects humans. It is round with an envelope, has RNA and N protein inside, and spike proteins on the outside. This is what attaches to our cell receptors to infect us. To infect our cells, the virus spike protein attaches to the H2 receptor on our cell surface. Then the virus enters the cell and releases the RNA by fusing with a vesicle that surrounds it. The virus then hijacks the cell and uses the cell's machinery to translate its own proteins and build more viruses. The newly made viruses are then released into the rest of the body to infect more cells. However, there are other cells, antigen-projecting cells, that engulf the virus and then display pieces of it on the cell surface. This is how the immune system reacts. These antigens activate helper T cells, which leads to a domino effect of immune response. T cells enable B cells to make antibodies. B cells can prevent the virus from infecting cells. They can also mark the virus with antibodies for other cells to destroy. Cytotoxic T cells are killer cells. They kill infected cells. B and T cells can stick around for months or years, waiting to respond if the virus shows up again. When the genetic sequence was coded in January 2020, no vaccine candidates were developed at that time. By mid-April 2020, there were 86 candidate vaccines from all over the world to combat this deadly pandemic. We can use the immune response to develop a vaccine to protect people from this deadly virus. Scientists are currently working on 4 main types of vaccines. The virus type, which can be inactivated or weakened. The viral vector type, which can be replicating or non-replicating. The nucleic acid type, which can have DNA or RNA. And the protein-based type, which can have a protein subunit or virus-like particles. The weakened or inactivated virus vaccine is one that can elicit immune response. The vaccine is weakened by passing it through animal or human cells to develop mutations that render it harmless. The virus can be inactivated using chemicals or heat. A weakened vaccine allows the harmless virus to replicate in cells, be consumed by antigen-presenting cells, and elicit an immune response in the same way the virus would, but without causing harm. Viral vector vaccines can be replicating or non-replicating. These use viral vectors that replicate in cells. In this case, the gene for the SARS-CoV-2 spike protein would be placed in a viral vector, and the cells would replicate the vector. The same immune response would be produced in response to the spike protein as with a viral infection, but it would be safe. Nucleic acid vaccines either use DNA or RNA encapsulated in a lipid coat for protection. Electroporation is used to make the cells permeable and uptake the genetic information. Cells make viral proteins, and a safe immune response is elicited. Finally, protein-based vaccines use viral subunits or virus-like particles. These subunits or particles are also uptaken by antigen-presenting cells, and an immune response takes place. These vaccines may require multiple doses since the parts don't self-replicate. Many of these vaccines are already being tested on cell lines, animals, or even humans. Scientists all over the world are working together to quickly make a fast and safe vaccine to curb this pandemic and save lives.